Thank you, Anna, for the kind introduction. As you said, I'm Miguel from the pathogenomics group at the I2SysBio in Valencia, and I'm going to tell you about, indeed, VIPERA and MIPERA, two modular frameworks for high-resolution tracking of intra-host evolution, whose development I've been leading as part of my PhD. So I'll talk about how and why we developed these two pipelines to study how two deadly pathogens undergo genetic changes during the course of an infection. Both take as inputs high-throughput sequencing data and produce a comprehensive report that is easy to interpret while keeping the raw results file, the intermediates file, tidy for downstream analysis. Well, I'll first introduce VIPERA, which we built for intra-patient evolutionary analysis of SARS-CoV-2, and I'll explain the rationale and the basics of what it does. Then MIPERA is built on top of VIPERA, inheriting some of its features. So then I'll talk about the updates to the biological assumptions to study MTB and the improvements that we introduced. Although these are a virus and a bacteria with, of course, very different characteristics, long-term or chronic infections can become very clinically important reservoirs of diversity for both under different timescales and selective pressures as well. Well, let's see. As you may know, COVID-19 was for years the main cause of death due to a single pathogen worldwide. It has actually taken more than 7 million lives and counting. And SARS-2 is a virus that has an error-correcting polymerase, so it's not so variable as other RNA viruses. However, it has had the chance to explore a lot of the genotype space. Since the start of the pandemic, there has been a continuous turnover of viral lineages. It's characterized with specific constellations of mutations, as you can see in the tree. And just in 2021, 3 variants of concern emerged and took over the last. Now, everything is Omicron recombinant, but that's a different story. What's important now is that variants of concern are often preceded by branches, or were often preceded by branches with an increased substitution rate. And our hypothesis is that chronic infections play a big part. So immunocompromised hosts may not be able to clear out completely the acute infections, which can lead to long-term infections where the virus has less adaptive restrictions. And indeed, there is a trend of detecting mutations that are typical of variants of concern in these chronic infections. So, it's key to understand long infections to understand how the virus evolves. Regarding MTB, well, after the worst part of the pandemic was over, Mycobacterium tuberculosis, MTB, went back to being the single most lethal pathogen worldwide like it was before. And for this bacterium, the epidemiology and the organism itself looks very different, as I said, but there are some parallels in short-term evolution. So just as chronic SARS-CoV-2 might seed future variants of concern, sustained MTB infections under incomplete drug pressure can generate resistant genotypes that then seed multi-drug and extreme drug resistance outbreaks or transmission clusters. Here, the key is that long-term infections are clinically relevant reservoirs of drug resistance. So, well, and also some evolutionary mechanics are actually somewhat similar. Both are slowly evolving relatively, both with limited to no recombination. So in short, there are different selective pressures, immune evasion and drug resistance for these two organisms, but the same question underlying: how long-term infections drive evolutionary trajectories. So what are the tools that we have available for genome surveillance? Well, there are ongoing efforts in standardizing human pathogen analysis focused on the population level, so between hosts. Here are some examples for SARS-2, for example. But there's actually like a competition standards in some cases, but this is not our case. There is a lack of user-friendly, reproducible, modular tools to analyze and report on complex processes such as the within-host prolonged infections. And also the approaches intended for these kinds of analysis tend to disregard variability at intermediate allele frequencies. So basically, this is what it looks like for us. Our aim is to start filling this void, and more specifically to develop tools to track how pathogen populations change within a single host over the course of infection, incorporating all genome variability from high throughput sequencing data. This is our code name, stands for Intra-Patient Evolution Reporting and Analysis, VIPERA. So let's start with VIPERA. As I mentioned, it's available right now on GitHub. Okay, so just a clone away. It has basically three main focuses. First, modularity. That's why it's implemented as a Snakemake workflow using open languages like Python and R. Snakemake is basically a great workflow manager that allows us to run each piece of the analysis, which internally are called rules. So to run it independently if needed, and import each piece into other workflows independently as well to reuse code. This will be relevant later for MIPERA. Second, there's reproducibility. So we achieve that basically using Obtainer, a container engine, to create a container itself with everything needed to run the software. And also we use Conda, which is a package manager, to automatically set up the environments we need for each analysis. So the idea that's frictionless, the setup. The third focus is deployability. We favor a simple way of executing this workflow via Slurm in a high-performance computing cluster. So the workflow requires just map reads and a genome, so a BAM and a FASTA for each sample, as well as their collection dates. And the main output is a self-contained report in HTML format that aggregates all the analysis. So intra-host variance, allele frequency trajectories, covariation clustering, evolutionary rates, and more. I'll talk about it later after the workflow overview. But as a plus, we've made efforts to make it user-friendly, semi-automated, and at the same time, transparent to the user. So in other words, we want to enable inexperienced users to run it easily on any environment out of the box, while keeping a clear record of where each result comes from in the workflow and allowing advanced users to inspect and maybe tweak if they need to every step of the workflow. So as a Snakemake workflow, we've structured the rules, each piece of the analysis in domain-specific snake files into main blocks, the core block and an extra block. I mentioned earlier, but very briefly, a rule is just a recipe for a specific file given a series of requirements, much like in GNU-Make for those familiar with it. The difference is that in Snakemake, these requirements can include other files, configuration parameters, Conda environments, etc. So this pattern makes rerunning an analysis or modifying with different settings really easy, because the Snakemake engine takes care of just organizing everything, which rules to execute, in what order, and if possible, if they can run concurrently, they do. So, the core block includes basically organism agnostic rules, and these are packed in a metasnake file that it is easy to import elsewhere. I'll show you later when I'm talking about MIPERA. And they include stuff like rules for fetching data from public databases, to process sequences, calculate metrics for molecular evolution, perform ancestral sequence reconstruction, the most recent common ancestor of the infections, calculate the dissimilarity between samples, perform variant calling against the ancestor to get just the intra-host variants, or characterize the sequencing quality of each site. Then there's these organism-specific rules from SARS-2, for SARS-2. These include fetching a sequence dataset that works as population-level background for some analysis, SARS-2 lineage demixing, typing, and the final report itself, which is indeed presented in SARS-2. So again, everything is designed from the start to be as portable and reproducible as possible, including the configuration parameters themselves. I mentioned that it's easy to keep track of a file and of any file and any configuration provenance by design. But at top of that, configuration is set on two levels. First, a global configuration, and also, apart from that, a per dataset configuration. In practice, this translates to having one file with sensible default parameter values that is very easy to edit. For example, with the evolution model, tree settings for Bootstrap, variant calling settings, the mixing settings, etc. Then, in a different file, there's it's in the same format, but it just has dataset specific configuration. For example, the output directory, sample names with paths to the required BAM and FASTA files, and sample dates. So the idea here is that there's no need to keep track of command line arguments. Everything is saved to disk and can be maybe shared or published like a simple file instead of a command line command. So let's start looking at how results look like. First, aside from the report, running the pipeline provides a well-organized results directory, as I mentioned, with subdirectories for each workflow domain. For example, phylogenetic tree inference, sequence alignment, demixing, population context, etc. Here, I'm showing just some important files as well. The variance table contains intra-host variants with the frequency and mutation functional annotation called against the ancestor after quality filtering, which is basically the center of most analysis. And now in the report, there's two blocks of results that translate directory. First, an aggregate of analysis for detecting whether there is a longitudinally sample infection or not in the input dataset, which we'll see in the next slide. This is very relevant to interpret the second block adequately, of course, which is the actual tracking of intra-host evolutionary dynamics. So here's how the header of the report would look like. It's made with Quarto currently. So just know that we published the workflow as a research article. We validated it using a positive and negative control dataset, and it worked really well. So I'm just going to show you some results of applying VIPERA to a real-world case study, the first time it was published. So the first block is a series of analyses for detecting whether there is a longitudinal sample infection or not, discarding co-infections, contaminations, as I mentioned, using an independent set of genomes fetched in the context submodule that I showed you earlier as a population level background. So the assumption is that serial samples from a single host infection, so the case study, will show lower genetic diversity than the independent viruses drawn from the broader population. And this broader population provides a background against which the workflow assesses automatically whether the target dataset is monophyletic or not, so if it forms a single clade or not, which is indicative of what I just said, that it's a single virus, no contaminations, etc. Then, by taking random subsets from the broader population, calculating the nucleotide diversity and comparing the distribution against the internal nucleotide diversity of the case study, the vertical line, we can do just a hypothesis test to address this statistically as well. And well, these two representations are very clear examples of what a long-term infection by a single virus would look like. So after confirming that the dataset came from an original single infection, there are some analysis of evolution. I already said, but keep in mind that all nucleotide variants or mutations here are called against the inferred ancestor. So via the ancestral sequence reconstruction under maximal likelihood that I'm showing denoting there. So all analyses consider variants that emerge during infection. So without going into too much detail, the workflow reports an overview of the intra-host variants of each sample in chronological order, the classification, SNP, indel, synonymous or not, etc., over the whole genome, and also the regions with a higher than usual proportion of polymorphisms. Then, building on this data, on this variant quantification, VIPERA also selects and reports variants with a high correlation between their relative frequency and time. And that means that here we see alleles with a significant upwards frequency trajectory over time, which are the most interesting clinically. Then substitution ratios, DN and DS, over time are also reported, which help track shifts in adaptive pressures within the patient. Importantly, here we were able to improve the estimation of the evolutionary rate using variants at any frequency. The usual approach is just using the fixed variants above a frequency threshold, typically above 80%, for example. So building then a consensus sequence and inferring a phylogeny based on this to serve as a basis for the estimation. So, well, under a simpler evolutionary model, our approach improved the fit of the molecular clock regression, and well, for SARS-2, and as we'll see later for MIPERA, this holds well under a variety of biological settings and, well, in MTB as well. So we ended up publishing the tool and our findings, and soon after we continued to work to extend this framework to other pathogens, starting with MTB and MIPERA. Since we had modularity in mind from the start, we've been able to accelerate development by reusing as much code as possible. And since many rules from VIPERA come from MIPERA with an M, come from VIPERA with a V, we have been able to keep maintaining VIPERA while we extend it, releasing new versions regularly with fixes, upgrades to the software, and also to the documentation. In fact, I'm very happy to say that some of them have come from user input, so from researchers that contacted us with suggestions or questions. So please don't hesitate to contact us if you have ideas or problems to solve. So yeah, anyway, although there's a lot of inherited code, around 2/3 of the rules are exclusive to MIPERA. And among the technical improvements we've implemented, there's indeed overall performance improvements. For example, we were able to solve some bottlenecks by switching to compiled rules, by using compiled software in some rules, and increased parallelism. Then there's also a clearer and interactive report, the detection of variants on the ancestor itself, a wildcard-based setup. This is key to of variant colors that will make it very easy to add new ones in the future, and also, for example, do more complex stuff such as aggregating results from multiple workflow branches, for example. I try to be as future-proof as possible. But now let's see a more applied overview of MIPERA. So MIPERA is an ongoing effort that builds more or less on the same grounds as VIPERA. Well, the logo and the manuscript are works in progress, but the aim here is to automate evolutionary analysis, as I said, of long-term MTB infections. The required inputs and outputs are the same, but crucially, now we're focusing specifically, well, especially, let's say, on an additional block, which is characterizing antimicrobial resistance over the course of an infection. Of course, it's not the only addition, as I'll show you next. So again, MIPERA is structured in domain-specific snake files with two main blocks. This time, one of the blocks comes from VIPERA itself directly, thanks to Snakemake's native module import capabilities. The syntax is right there. So it's as simple as importing the core metasnake file I mentioned earlier, the core one, from the GitHub repository. And in the second block, we just extend the functionalities. So the most relevant ones are variant calling, which we use different colors than in SARS-2, BAM, BCF file handling, genome alignment, static visualizations, which are here the minority, as I said, because it's mostly interactive visualizations. Then there's the antimicrobial resistance annotation, per codon clustering of single nucleotide variants into multiple nucleotide variants, which is very relevant for annotating the antimicrobial resistance correctly. Then there's an analysis of polymorphism hotspots, which is a bit more sophisticated than in VIPERA. I'll go in more detail in a moment, and the code for generating the report. Importantly, due to the significant increase in complexity, VIPERA now includes rules with sentinel targets, and the idea is that it makes it easier for users to obtain just a specific block of results. It's a way of requesting, for example, I want just variant calling using Freebase and none of the downstream analysis. You can do that now very easy. Again, this makes for a just easier user experience. So in VIPERA, let's go with the results. The blocks are simpler. There is just tracking intra-host evolutionary dynamics. Since our manuscript is in progress, I'll showcase just some of the new analysis and visualizations implemented in VIPERA using a dataset from the literature. So this was the first document case of extremely resistant tuberculosis that evolved from a susceptible ancestor, which is very interesting for our case. So to try to recapitulate their findings using our semi-automated tool. So first, we're greeted with an overview of the available sequencing data and a chronology of the, a chronology and also a minimum spanning tree over a principal coordinate analysis. So multidimensional scaling that is based on pairwise variant dissimilarities between samples. These pairwise dissimilarities are the ones that are used, I showed you, for the molecular clock regression. And this helps visualizing just an idea of the structure of the dataset. Then the overview of the nucleotide variants in VIPERA is richer, more intuitive, and includes an interactive plot that is GPU accelerated via WebGL to visualize this way thousands of variants without losing responsiveness. Here you can zoom in and out or pan the view over the whole genome length. So if you're interested in a gene, you can just go there. On hover, as you see here, the functional annotation of each variant can be very easily visualized, as well as its location and so on. Then in the lower panels, there's a more sophisticated way of highlighting polymorphism hotspots on these regions, as I mentioned earlier. So regions include both features in the first strip. Here it's genes. And in the last one, there's non-overlapping windows of the same size. Here it's 1 kilobase. And briefly, we just fit a Bayesian hierarchical beta-binomial model per region, accounting for overdispersion in polymorphisms across regions, which can be attributed to lots of evolutionary effects or non-evolutionary effects. And for each, the number of polymorphic sites out of the callable sites, which are all pulled across the whole time points, is modeled with the priors centered on the empirical genomic wide rate of polymorphism. Then the reported regions that are in the report, well, are those with a significantly higher rate than the genome-wide baseline, and we enforce just a false discovery rate threshold for those. And on hover, the report displays, well, these results, the details about the region, the observed counts, the inference results. Now, a key addition to VIPERA is the identification of variants that are associated with antimicrobial resistance. And to do that, the workflow automatically fetches the WHO catalog of antimicrobial resistance, AMR mutations. It processes it and annotates for all samples and available drugs all the intra-host variants that can be found within 5 categories that you can see there, through association to through lack of association. And in our literature dataset runs, that's a figure from the paper. We have found perfect matches using our automated approach. It recovers all the reported emergent resistances and sometimes variants that were uncharacterized at the time or not fixed, so not so below the allele frequency thresholds that the research article used originally. And in addition, the workflow reports the trajectories in time as well as their presence or absence that we saw in the last slide. So paired with knowledge of drug regimens of the patient, tracking this can really be very informative for both evolutionary biologists for downstream analysis, but also for clinicians that are tracking a specific problematic case. Maybe. Then, based on these AMR and also non-AMR trajectories, MIPERA includes two covariation analyses that aim to detect groups of variants with parallel of or opposing changes in frequency. So the first one uses hierarchical clustering, and the second one uses network-based community detection, here restricted to variant pairs with a coefficient, which is the edge weight that is above a threshold. These results can, for example, point to candidate mutations that may be functionally or maybe epidemiologically linked, although covariation can also arise from hitchhiking or clonal dynamics rather than through epistasis, but variants close to AMR mutations could be of clinical interest anyway. So to wrap up, I mentioned that just the last of the results, I mentioned that a key addition of VIPERA was improving the evolutionary rate estimation by incorporating variants at any frequency via the neighbor joining tree, using a neighbor joining tree built based on the pairwise dissimilarity metric we developed. Usually the standard approach relies on fixed variants only under a specific, so explicit model of evolution under maximum likelihood or Bayesian approaches and so on. So MIPERA, as opposed to VIPERA, now reports both trees, so both approaches to help assess users the broader picture. But I have to say that even under a simpler evolutionary model, our approach really yields a better fit in this setting as well. So in both SARS-2 and tuberculosis intra-patient cases. So as take-home messages, these are modular and user-friendly tools to automate analysis of intra-patient longitudinal samples, enabling easy downstream analysis. Incorporating all allele frequency data can yield more informative insights into pathogen evolution, as you just saw. And our framework has the potential to eventually become a versatile platform to analyze the evolutionary dynamics of any organism. So I'd like to acknowledge our funders and our group members present at past, especially Jordi and Yuna, both great bioinformaticians who contributed a lot to these tools as graduate students, and also the connection for the opportunity to present here. I'm happy to discuss either here or via email or DM. Thanks.